Brain. Anyway, our first speaker, this is on neuroscience. We have the first two speakers are remote. The first speaker is very well known to most of us, or many of us, Robert Lawrence Kuhn. He's been the producer and star of Closer to Truth, which has been running on PBS and now YouTube for I can't remember how many years. Maybe he'll remind us. And he's actually interviewing many people here remotely. And so those interviews will be coming out on the show Closer to Truth in the future. And he wrote a paper this past year that kind of caught fire among the consciousness aficionados around the world about the landscape of consciousness, where he detailed, I think, 275 theories of consciousness. And many people think there's too many theories, and some people think there's not enough because we probably don't have the right one yet, although I disagree, but anyway, that's just me. But anyway, he's going to speak to us about landscape of consciousness, comprehensive, authenticated, and up-to-date. Robert Lawrence Kuhn. Take it over, Robert.
Stuart, it's great to Thank you, everyone. Good morning from New York, or good afternoon in Barcelona. Stuart, it's always great to see you. We first met on Closer to Truth in 2003, and Stuart hasn't changed a bit. So I may have gotten older, but he hasn't. So we've had a great time, and we've interviewed Stuart many times over this period. So I want to talk to you today about the landscape of consciousness. About a year ago, as Stuart said, I published a paper on theories of consciousness, and it is honestly the product of a lifetime. It has over 225 theories in 10 categories: materialism, non-reductive physicalism, quantum and dimensional, which I put together, information, where information is ontological, and then the ones you know, panpsychisms, monism, dualisms, idealisms, and then two final categories on anomalous and altered states, which are not theories of consciousness per se, but affect theories of consciousness. And then a last category called challenge theories, which are orthogonal and different approaches to the subject. The paper is called The Landscape of Consciousness Toward a Taxonomy of Explanations and Implications. I specifically say not the, because there are many different kinds of taxonomies that one can do. In the article, what I seek to do is to provide an organizing framework for all of these diverse theories of consciousness and to explore their impact on big questions. What I try to do is I try to collect and organize the totality of humanity's knowledge and wisdom about consciousness, certainly emphasizing all the science, of course, which are largely the materialist theories, but not restricted to science. And some of these categories blended, and some of them are clearly non-scientific, and some of them are sort of in the middle. Now, I have two central theses that I'm trying to propagate. The first is that understanding consciousness at this point should not be limited to selected ways of thinking or knowing, but should rather seek expansive yet rational diversity. And the second point is that issues such as AI consciousness, which everybody's talking about, virtual immortality, meaning, purpose, or value, even life after death, cannot be understood except in the light of particular theories of consciousness. So whatever your particular theory is, it will have implications for all of these big questions. And to address these big questions without having an underlying theory of consciousness is incoherent. And if you don't have an underlying theory of consciousness, you actually do. You're just not aware of it. So I want to make everyone aware of whatever their theory of consciousness is, and then address these questions. Those are the questions I addressed in the paper, which is online. a landscape of consciousness. But other topics also are wholly dependent upon which theory of consciousness you choose, which I didn't deal with, which I could in the future, like free will, personal identity, what animals or life forms are conscious, those kinds of questions. But today what I'm doing is not reviewing the paper from the past, but introducing and announcing here at the Science of Consciousness Conference the Landscape of Consciousness website, which has been in development for a year since the paper. It's a dramatic expansion and extension of the article, and there are now about 300 theories in still the same 10 categories, and one of the categories, materialism, has 12 subcategories. The reason materialism has so many subcategories where the others do not is that if you have a materialist point of view, you are able to subdivide that more into different kinds of scientific ways of thinking, which I will go through with you in a moment. Whereas in the other categories that are less scientific or non-scientific completely, you can give the theory, but you can't make much progress beyond that for the reason that it's not in the same sense scientific. So what I want to do is take you through the website, and I have certain slides from it that I'll show you. But I'll give you, and it's better for you to look at it on the computer itself. It's just released literally yesterday in beta form. So if there are bugs, we're going to keep it in beta for 4 to 6 months. So we're asking the consciousness communities, broadly construed globally, to check out the website, give us any suggestions of any kind. You find bugs, let us know. It's loc.closertotruth.com. LOC for Landscape of Consciousness, but the URL is loc.closertotruth.com. As Stuart said, Closer to Truth is a U.S. public TV web series, website, and YouTube channel. I credit Peter Getzels, who's the co-creator and director, producer of it. So on the Landscape of Consciousness website, we have presenting all 300 theories in different forms. Each theory has its own page, and the description of it is anywhere from 400, 500 words minimum to maybe 1,500 words maximum. And most of it is taken from the individuals themselves. And what we say about the website is 3 things. One is that it's comprehensive, and I think you would give me that. If it's 300 theories, you would think it's comprehensible. There are a lot that we've rejected. It's authenticated, and what that means is that with many of these theories, not most yet, the actual theorist has said that this is an accurate description of his or her theory. And that took some effort. In the original paper I did, since I knew personally from Closer to Truth 30 or 40 or so of the theorists, I sent it to them ahead of time, and they gave me their feedback. Most of them were very gracious, said it's good, maybe making a few changes. A handful made significant changes, and I apologized. I said, look, I'm sorry I got it wrong. They said, no, you got it right, but that's what I thought 2 years ago. You were working off my old papers, here are my new papers, and they changed it. So what we're aiming for is to have as many of the total theories authenticated by the author, and when it is authenticated, there's a little box on the theory page that says verified. We use the word verified, and it gives who verifies it and when they did it. And usually it's the author themselves. Obviously, for some people who have died, what we do is we will find the leading experts who know their theories best to authenticate it. So it's comprehensive and authenticated, and we say up to date, because it is always open for input, updates, revision from the theorists themselves. It's not a wiki. People can't do it themselves. We curate it completely ourselves. But anytime whose theory is there, they want to add a reference, whatever they want to do, this is the source. We want this to be the source where you can read, it's much more than an abstract about the theory, but much less than a full research paper or an big encyclopedic article. 500 to 1,500 words, authenticated, comprehensive, and up to date. So I think you can see the screen, and this is the opening screen where we have it, and it has 4 ways in which to visualize and access all of these theories in these categories. And let me start, and I'm not able to do this on live clicking because of the way the system is working, so I have static images, but you can get it yourself. So the first one I'll show you is what we call the consciousness map. And this map, the design was created by Alex Gomez-Marin, who has been working with me very closely on the project and is speaking at the conference. He created the image, and it's populated. And in this case, it's only 1 or 2 words just to get it all on one map. We'll see it in a different form as we go through. So here you can see the first category, materialism. And as I said, we have the 12 subcategories that are under it. And I'll go through those later. And then non-reductive physicalism, quantitative and dimensions. And each one of these words represent an individual theorist or their theory. Information, where its information is ontological, panpsychisms, monisms, dualisms, and then idealisms, anomalous states, and the challenge theories. And the challenge theories, you can see some names that you know: Tom Nagel, Colin McGinn, who are, Mysterianism, different approaches to the theories. So that's one way to see it, is through this consciousness map. Now let me go to another way to visualize it. So this way is a grid. And unfortunately, I'm not able to use the sharing but this is all active. So across the top, you see the major 10 categories of the landscape. And each one of these then will have a grid. When you click on it, it will have all the theories underneath it. When you click on the theory itself, it'll give you a short summary in 2 sentences with a link saying, here's to go to the full discussion, which is, as I said, 500 to 1,500 words. For materialism, it has subcategories. So you have the 12 subcategories here that you can read, and I can read them. The philosophical versions, which are, things like that you'll be familiar with: functionalism, supervenience, mind-body identity theory, emergence, those kinds of things in philosophical. Eliminative and illusion, you're familiar with that. Keith Frankish, Dan Dennett, others. The one I've highlighted is the neurobiological, which is the longest. And these are theories from Gerald Edelman to Crick and Koch's neural correlates of consciousness. You can skim them. Global workspace, many different theories that are strictly neurobiological. And the next one is electromagnetic fields, in which you would see a similar thing. And then computational and functionalism. For example, the Lenore, Emanuel, and Avram Blum, who are computer scientists, have a very elaborate computer science approach. And then the other categories, we have homeostatic and affective, emotional things, Antonio Damasio, etc. Inactive and embodied and inactive theories, Evan Thompson, among others. Relational, and then first order representational, higher order representational, and then language relationships, and then phylogeny. These are all the subcategories under materialism, and each one would have 5 to 15 different theories that you can access. So now let me go to another one. So that's 2 ways to see it. And this one, unfortunately, I'm not going to be able to. And this gives a linear picture of all 300 theories. And so you go to, this is called Browse All Theories. So when you go to the website and you see the button on the homepage, first panel, Browse All Theories, that's what this is. And this is a list of literally 300, if I could scroll it, and you just see materialism with the first 2 or 3 subcategories under it. And each of these names, which are now the full name, as opposed to one word on the map, because that's all we can get. But this has all the names, and you click on each one of these and you go to the major theory. The last one I want to show you is sort of a new one that is really interesting. Let me just see if I get the right picture of it. And this is a scatter graphs of the of most of the theories, not all, created. The visual image and was created by a fellow computer scientist in Turkey named Deniz Cem Angdalu. And I give him credit, and we work together as partners. And he has a number of colleagues who work in AI, a deep mind. And what we took is all the theories. Well, we had a sub. We didn't take all 300. We took about 200. And we fed it into Gemini 2.5 Pro and asked it about 5 different characteristics: the complexity of the theory, the connections between theories, the popularity of the theories as judged by hits on Google or hits on Google Scholar. Those are 2 separate things, and some other characteristics, and we embed them within these charts. I just commend you to go to the website and look at interactive visuals or interactive images. There's a whole page. There are 4 of these. Let me see if I can show you another one. This is a page, the one I'm showing you shows the interconnection between the theories, which ones connect to each other. And it's not perfect, and we're looking for feedback, but to do it through Gemini 2.5 gave us some standardization, which we wanted to do. Let me see if I have another image. No, that's not working. This image is really one of the best. It didn't show up here, but you'll see it. And it also plots the order of magnitude of where the consciousness occurs. In other words, is it all the way at the left hand would be, at the quantum level, all the way at the right hand would be cosmic consciousness, and the middle would be at the brain level. And then in the y-axis, it was materialism to an idealism. And so you have the scatter picture of it there. So once again, let me see where we are. And so we have these 4 separate ways to access the theories, but the main theory page I'm not showing, but that you easily can get to. So we ask everyone to go to the site. Again, it's loc.closertotruth.com, and explore it. Feedback. My email is there. Anxious to get your feedback, get your participation. Any theories that are left out, please send. In fact, since the original paper was put together, I've gotten literally hundreds of theories, of which we've chosen about 60 to add to the original number, and I think it's much richer for it. Theories we get wrong, or theories that are misplaced in terms of the taxonomy, we want to hear all from your feedback. So please tell associates in the consciousness communities about the site, and we look upon it to be a community resource for everyone. We're committed to make the effort with our team to maintain it and to work with all of the theorists themselves to make sure it's always comprehensive, authenticated, and up to date. So thank you very much, everyone, for participating. I need to get back to our interviews that we're doing to promote the science of consciousness. And so I thank you once again.
Thank you, Robert. We'll put out the website to our email list and broadcast it. And thank you for doing that. And we look forward to see it evolve and over the future. And hopefully, well, I don't know if it'll come to a conclusion or not, but that's not the point. So thanks for doing it. And thanks for being here. And I'll talk to you on Friday afternoon.
Yeah, look forward to it, Stuart. Thanks, everyone.
Okay. Another round of applause. Okay, our next speaker is also online, and he just applauded also. He is Earl K. Miller, a professor of neuroscience at MIT. There he is. And he's also a musician and a bass guitar player for a band called Pavlov's Dogs with a Z. And he's a great guy, and he's going to talk about consciousness emerges from neurodynamics. Earl K. Miller, take it away, Earl. Thank you.
Stuart, thank you very much. It's great to hear from you again. It's great to give a talk to this meeting, and I thank you for the invitation. And Robert, that was very, very exciting. I'm going to jump right in. I'll warn you that we're about to get very, very materialistic. I am a neurophysiologist. I study the neural mechanisms of cognition, and my thoughts about consciousness sort of came bottom up by letting data show me where to go. So let's just dive right in. So many biological theories of consciousness have one thing in common: the formation of large mesoscale networks or ensembles that stretch apart across large expanses of cortex. And here, just for illustrative purposes, I've illustrated the global workspace theory. And this formation of large networks that sort of organize across the cortex makes sense because conscious experience is a unified whole. We experience sights, sounds, everything all fused together into one experience of consciousness. But the question is, how does the brain organize hundreds of millions of neurons into these large-scale networks? And this is another way of asking, how does the brain think? Because cognition is organization. Goal-directed behavior, thinking, what you're doing now is your brain, your cortex is organizing hundreds of millions of neurons to focus on one thing, hopefully me and what I'm talking about, and thinking about that. And it's all being coordinated in a way that's directed towards your current goal at hand. And this question of how the brain organizes itself to produce goal-directed behavior has been the question that has guided my research as a neuroscientist for the past 30 years. And the traditional view of how this works, the view we inherit from the 20th century, is that the brain computes and stores information by modifying network connections. This may be familiar to you because this is how AI works. AI is modeled after this. You have connections that between units that store information, and when you want to predict what the next word should be, you shift around connections to find that prediction. So connectionism. And we used to think that every neuron is specialized, sending these electrical impulses, which we call spikes, these little brief electrical impulses through synapses. And these are analogous to the ones and zeros of digital systems. Short way of saying that the traditional way we used to think about the brain is that the brain neurons were like gears in a clock where every neuron had one function. And it was kind of like an elaborate telegraph system where brains worked by changing connections and sending impulses along these connections. And just to illustrate this, here's a simple example that comes from the inspired by the work Nobel Prize-winning work of Hubel and Wiesel. Imagine you have this image. You're looking at this image on your retina. So you're looking at this image, and there's images on your retina. Yeah. Well, that's weird because I was advancing the slides. What about now? Are we advancing now? I don't know what to say. Hang on a minute. Let me try resharing. think what I did is rather than, we'll get this working, I assure you. Yep. Yeah, I am in slideshow mode. I'm trying to get the meet screen back. Hang on one second. Okay. Let's do entire screen. This should now work. Okay. How's that? All right, awesome. Sorry for the amateur hour, people, but not used to meet. Anyway, the traditional view of the brain is this connectionism, and here's how we think it works. How we used to think it works. So you have this image on our retina, and then at the first layer of processing, you have these neurons that are specialized for detecting edges. And then the next layer of processing, you have these neurons sum together to produce longer edge detectors. And then they sum together to produce corner detectors. And then you sum, you have millions of neurons, and eventually you get complex shapes, and you have lots of neurons in your brain. So you summate enough of these specialized responses, and you get peace, love, and understanding. That's the way we used to think about the brain when I was a graduate student in the 20th century, and some still do, quite frankly. But our work led to a critical insight that neurons are not like gears. Neurons are not specialized. They're multifunctional. So here's the traditional view of neural coding that I just presented to you. We have neurons they detect single features like form, color, or motion. Then you have summation, so you might get some neurons that detect some related features. So some neurons might detect red, round, and ball, and other neurons might put them all together into the image of a red ball that's rolling to the right here. But our work in studying how the brain organizes itself revealed a new population of neurons that was previously undiscovered, these multifunctional mixed selectivity neurons. And what these are neurons that combine many different features of all sorts of different information into unique combinations. And these combinations don't seem to have any single combinatory principle or function like red rolling ball. And importantly, they're multifunctional. They adapt their signaling to play different roles in different situations. The neurons don't do one thing all the time. They do different things in different contexts. And what we found is that these mixed selectivity neurons are actually critical for high-level cognition. And the reason is the math shows my colleague Stefano Fusi, Mattia Ragati, working with Melissa Warden, who provided the data, we showed that these property of mixed activity adds the horsepower to the brain needed for high-level cognition. It's a substrate for combining a wide range of information. These neurons, because they have access to so much information, they can combine arbitrary information into new things. And this is what human behavior is all about. Our brains did not evolve to sit in front of computer screens or sit in an audience and listen to a speaker. We can do arbitrary things. So these are a substrate for pulling linear information, into arbitrary relationships. And these higher dimensions provided by these complex neurons, they provide a higher dimensional representational space that makes the brain smarter, gives a more computational power. And it increases the brain storage capacity, these multifunctional mixed activity neurons, because they distribute memories across many neurons rather than into a few specialized ones. And for all these reasons, this leads to exactly what the kind of things we need for what we think of as high-level cognition: fast learning and cognitive flexibility. But there's a problem. This property of mixed selectivity means that our previous model, where the brain is like a elaborate telegraph system, is actually more elaborate. It's more elaborate than that. The cortex is like a multitude of overlapping networks, all sharing synaptic connections. This means that neurons don't just do one thing, they do many different things, and they shift their networks, which networks they belong to, depending on the context. They shift their function. And what this means is that connections, the kind of things that AI was modeled at, and the kind of things that dominated our view of the brain in the 20th century, this cannot be the whole story. And the reason is because intelligent, goal-directed behavior requires the large-scale coordination of millions of neurons, as I mentioned at the beginning of my talk. And importantly, this is self-organization. Our brains don't just react to inputs from the environment. We act on the environment. We organize our own thoughts and actions around our short and long-term goals. And that's very different from the way AI works. AI only works when you give it an input and it gives you a response. AI does not think between inputs. Your brains think all the time. And of course, we have this flexibility that I've been talking about. This is rapid switching. This is intelligent goal-directed behavior. What you're doing now depends on rapidly switching your mental sets depending on changing situation, changing demands, new contexts. And it just seems impossibly complex for an executive control system, for a brain to control itself, relying just on connectivity alone, like AI currently does. And another thing that many neuroscientists don't talk about is spiking in synaptic transmission is relatively slow. If I show you a visual input, if I show you a picture on a screen, it takes about 100 milliseconds for that spiking, carrying that information to reach the front of your brain. 100 milliseconds is an eternity in brain processing. So what's the solution? If this connectivity story doesn't work, it's too complex, too slow, what's the solution? Well, the solution may be the most obvious thing the brain is doing. Namely, your brain is producing oscillating electric fields, so-called brain waves. And this is something that I'm sure many, if not all of you, are familiar with. These are EEG scalp recordings down here in the lower right of the screen. If you put electrodes on your head, you'll see all these squiggly lines. What these squiggly lines are millions of neurons activating, deactivating together at different frequencies, anywhere from 1 hertz up to 100 hertz or more, or gigahertz, if you ask Stuart. And the thing about these brain waves is they're an emergent property. They're not explainable at the level of individual neurons, which was the focus of neuroscience in the 20th century. An example of emergent property here is on the right. Here is our crowd. Here's a sports crowd. And you see the wave is moving from right to left across the screen. If I would study each person in the crowd one at a time, like we used to study the brain, I could hear what each individual is doing and saying. I could hear the noises they make, but I can't see the wave until I study them all together. So that's why the wave is an emergent property. But importantly, these waves, brain waves, oscillations are organization. Electrical networks, when you hook them together, they will naturally oscillate. Ask anybody who's tried to build a radio. The circuits naturally oscillate, and you got to wind wires together to cancel out the oscillations. But evolution could take advantage of this. My guess is that early in evolution, nervous systems began to get complex enough to naturally begin oscillating. The neurons began to fire together and go quiet together in unison. And when you have an oscillating system, when these elements are oscillating together, that is organization. It may be a dumb organization, but it is organization. And evolution could take advantage of this, evolving mechanisms that push around the oscillations in order to control the system at this emergent level. And this is important because wave dynamics make control easy. Waves, unlike trying to organize a system at the level of hundreds of millions of individual neurons, which is computationally extremely difficult, wave dynamics are easy. They're computationally tractable. Wave functions are simple. If you know a wave's properties, you know its effects in the future and at a distance, because wave functions, that's exactly what wave functions tell you. And there is a reason, likely why at the quantum level, the universe works by using waves, because it's computationally tractable. And importantly, electrical field influences, these brain waves that was ignored in the 20th century because they weren't involved with synaptic transmission. These electrical field influences, brain waves, spread around the brain 5,000 times faster than spiking and synaptic transmission. So that sounds like because it's computationally tractable and the influence spreads so quickly, it sounds like a pretty good way to organize the brain to me. And importantly, there's something called representational drift. Anybody who studied individual neurons in cortex will tell you that individual neurons are fickle, noisy, and unreliable. They come and go. They give a little burst of activity. They're quiet for a while. Then they give another little burst of activity, of the spiking activity. In other words, the cortex of your brain, all these neurons is like a giant orchestra. And the song is playing on, But each individual member, each neuron is only contributing a snippet of the song, then they're being quiet for a while, and then they're playing a little snippet of the song, then they're quiet for a while. So if you want to control the orchestra, you can't do it at the level of individual neurons because it's not only computationally extremely difficult, but individual neurons are not reliable players. So the brain needs to organize itself at this higher emergent level, not on the neuron level. And we've shown this is work from my colleague Dimitris Panosis at the University College London. We did something bold according to the old view of traditional view of looking at the brain. We ignored neurons and we studied the electric fields directly. And what we found is you could read the contents of thought, the contents of working memory from the brain's electric fields. And there's none of that representational drift, none of that fickleness at the level of individual neurons. You get a pure, constant, robust signal at the level of these electric fields. So that sounds like a pretty good way to study the brain. I can now get this pure signal without this fickleness problem. But importantly, we have to keep in mind that these electrical fields have a causative influence. The way it works, and there's this is just one review that talks about this, but there's thousands of papers on effect decoupling. And the way it works is these individual neurons start giving off these impulses, and they start the electric fields fluctuating around them. Then the electric fields begin to take over, and they have a bidirectional influence and then influence the underlying spiking. If you get enough spiking, the electrical influences they can start taking over and rollicking the sea and really taking over from the neurons, controlling the underlying activity of individual neurons in the brain. So think of neuron spiking the way, again, in the 20th century, we used to focus on neurons alone. But now I kind of think of neuron spiking as like buzzing your lips into the mouthpiece of a trumpet. The notes start there, but they fully form in the resonance patterns in the trumpet's body. And that's what I think is happening with neural spiking. It's like the neural is creating these resonance patterns in electric fields, and that's where a lot of the action and organization happens. And this just describes how electric fields can have an influence on the macro level, organizing hundreds of millions of neurons on a large scale. Dimitris and I also published a paper 2 years ago where we said, in addition, these electric fields, these oscillating electric fields can also have an influence on the micro scale of the brain. In addition to acting on this large scale, electric fields can also restructure the brain on the molecular level. Now on the left here is an example of, have you ever seen, if you pour a bunch of grains of sand on a speaker and you play a note, the sand will organize itself into shapes. That's what we think is happening, is these electric fields are oscillating and they're pushing around, shifting around the structural proteins on the molecular level in the brain, fine-tuning the networks of the brain to oscillate more efficiently. And that's what biology is all about, doing things with as little energy as possible, making things function more efficiently. But because these electric fields are carrying information, that means the information the brain is processing can restructure the brain on the molecular level. So I submit to you that this is one of the first molecules to mind theories connecting all these different levels of brain processing. And we've shown that different brainwave frequencies have a real functional role. So we found that high frequencies, brain waves at 30 hertz and above, these are high frequencies in the brain, they feed forward sensory inputs from the back of the brain to the front of your brain, the executive part of your brain that takes charge. And at the same time, the executive part of your brain does the opposite. It sends control signals back, and these control signals are happening at a lower frequency. And what these low frequencies do, they suppress, they control, they organize this high frequency gamma, so it filters and organizes sensory inputs and controls and organizes the brain. So the idea is that low frequencies control neural processing, and the high frequencies carry the contents. And this makes sense from a physics standpoint, because it's easier for low frequencies to control high frequencies than the other way around. So think of this, we've organized all these thoughts into recently to a theory we call spatial computing. And the way we think about it is these alpha, beta, low frequencies are providing the top-down control. They're like a stencil that the brain sends out to say, only these parts of cortex where the low frequency waves are not appearing, only those are allowed to become activated. And then the outside world, using this gamma, acts like spray paint that fills in the gaps between what the brain, between the parameters the brain sets. So it's a way of thinking of network space as a coding dimension. The brain uses spatial patterns of neural activity controlled by oscillatory rhythms to control which networks process information. And that's how your brain does computation. One of the ways the brain does computation. And this whole process here, we've formed modeling where the separation of content, the data your brain is processing from the control into different frequency spectrums, we've shown enables all the hallmarks of cognitive flexibility. Because you're separating content and control on different levels, they can each change without influencing one another, and you get rapid generalization and a highly flexible system. But what does this have to do with consciousness? Well, we've shown in a series of work I've done over the past few years with Emory Brown's laboratory, who's a MIT professor and a practicing anesthesiologist, that general anesthesia alters brain waves in fundamental ways. It doesn't just turn off your brain. It's maybe disturbing to know, because anesthesia has been around for over 100 years, that medical doctors don't really know why it makes you unconscious. They just know it does. And there's been this tacit assumption that it just sort of turns off your cortex, just your cortex shuts down. But we've shown it doesn't do that at all. It actually changes the brainwave patterns in your cortex. So here's examples of electric field recordings called LFPs, or local field potentials, from 4 different brain cortical areas. This is the front of the brain, the middle of the brain, that's frontal cortex, front of the brain, the middle of the brain, parietal cortex, middle of cortex, and auditory cortex. Sensory cortex, and these are all electric field recordings, and you see what you see, and here's the underlying spiking activity to go along with it, the individual pulses from single neurons. And what you see in the normal awake state is a bunch of high frequency radio chatter as these waves form and unform and organize at different levels. So you see a lot of stuff going on. But then we anesthetize our animal with propofol, commonly used anesthesia, and we see something very different. All this high frequency FM radio chatter is now being replaced by this low frequency delta hum, these 1 hertz electric field fluctuations, very slow electric field fluctuations that entrain the underlying spiking activity. Okay, so your brain just suddenly starts going slowly into these slow pulses. So that shows that anesthesia doesn't just turn off your brain, it dramatically changes the brainwave profile. But here's a mystery, is that what I'm showing you here on the right, with these slow 1 hertz so-called delta waves, this is what your brain looks like when you sleep during slow wave sleep. But I can do surgery on you under anesthesia, but I can't do surgery on you when you're sleeping. So what is the difference? Why is unconsciousness by anesthesia different from slow wave sleep, when, at least in terms of this level of brainwave analysis, they look the same? So how is anesthesia different? Well, the answer to this comes from a study we just recently published, where we found that the general anesthesia doesn't just shift your brain waves into these lower frequencies, it misaligns the brain waves. Normally, these brain waves are highly in sync with one another. They oscillate together. And after we anesthetize the animal with different anesthetics, we find that these brain waves become misaligned. They become 180 degrees out of phase of one another. So the brain waves used to be in phase of one another. If you can see my hands, they're oscillating in phase of one another. Then after anesthesia, they become completely out of phase of one another. And signals that are out of phase of one another, 180 degrees out of phase of one another, they can't transmit information, because what the oscillations are different levels of excitability, than high activity, low activity, excitability, inhibition. And if 2 brain waves are completely out of sync with one another, their excitatory periods, the part where they're really highly active, no longer line up with one another. And that could prevent the formation of these large-scale ensembles in cortex needed for consciousness. So we're now studying sleep data to see if sleep doesn't do this. If during sleep you get the slow waves, but they remain lined up, or anesthesia misaligns the brain waves. But what I find exciting about this study that we just published is that we found this effect in this study, we found it with 2 different anesthetics, ketamine and dexmedetomidine. And just recently, breaking news, we've also found the same thing with propofol. Now, these 3 different drugs, ketamine, dex, and propofol, they act on different molecular receptors in different parts of the brain. They act on different cell types. Propofol has inhibitory effects. Ketamine has excitatory effects. On the molecular level, on the detailed level, all 3 of these anesthetics do something very different. If you ask my reductionist colleagues, how do these three anesthetics produce unconsciousness? They would look at this and say, oh, it must be three different pathways, three different mechanisms, three different ways that these drugs produce unconsciousness. We're finding that all three drugs do this exact same thing. They do the exact same thing at this emergent brainwave level. You can't even tell where the data come from, which anesthetic was being used. And what this suggests is that this brainwave misalignment is a universal signature of unconsciousness that cannot be seen or explained in a more reductionistic level. And if we see a universal signature of unconsciousness that is common across all these different drugs that have different molecular actions, that tells us that this emergent level of brainwaves is really important for brain function. And it's a compelling argument, I think, that consciousness depends on brainwaves. Now, to see how this works in another way, these waves, these brainwaves I've been telling you about, don't just stand in place. They're not standing waves like a jump rope. They form traveling waves that move across your cortex, and like ocean waves coming in and propagating or dropping a pebble in a pond, and then the ripples move across the pond. These waves are constantly moving around. So what do these traveling waves do? Well, what we think they do is they organize underlying cortical activity. They organize the individual neurons to make them operate together. And what I've shown here, this is an example. Now I'm not talking about electric fields now. Now we're back down to the level of individual neurons. And what I've represented here in this figure is something called subspace coding. There's a mathematical representation describing how a population of individual neurons, in this case, is about 200 neurons, how they all work together. And what subspace coding is that I have recordings from 200 neurons, all giving these little spikes, these little impulses, all at the same. I have recordings from all of them simultaneously, and I can describe the patterns that these individual 200 neurons form by creating a 200-dimensional space and just plotting the activity, the spiking, the impulses of each neuron along each dimension. That's self-evident. That's just you're self-describing what the neurons are doing. So I could describe what these 200 neurons are doing with a 200-dimensional space. But now we ask the question, how many dimensions do I actually need to describe what these 200 neurons are doing? And the answer is you can do it in just 3 dimensions. 3 dimensions. That means spiking in the cortex, these individual neurons giving off their activity, is highly coordinated. Rather than being cortical spiking being like a bunch of insects buzzing in the night sky, spiking the individual neurons in cortex is highly coordinated, like a murmuration of birds moving across the sky. And what you're seeing here is, in this case, this is over the course of a memory delay. What these 3 different this is a task where the animal is holding 4 different pictures in working memory. And what the 4 colored lines are, the different pictures, and what you're seeing is processing evolving over time as the animal's holding this information in memory, the activity circulating around the cortex, but still the cortex is keeping all 4 different memories separate from one another. So this is actual neural computation at the level of individual neurons you're seeing in this highly coordinated fashion. So how does this organization take place? How does the brain, again, back to our original question, how does the brain organize this activity in this murmuration of birds-like fashion? Well, what we found recently, and this is a paper that hopefully is about to be published soon, is there's a direct correspondence between the movements of spiking patterns of neurons in this mathematical subspace coding representational space and rotations of traveling waves of neurons across your cortex. So what you're seeing on the bottom here is not spiking of individual neurons. They're electric field recordings from an array of electrodes, where each square is a different electrodes, and they're literally arranged in this square format. And the color is electrical potential. And what you see here, and this is unprocessed raw data, you see that there's a wave of activity that's rotating counterclockwise across the surface of cortex, rotating in a counterclockwise fashion across the surface of cortex. And that directly corresponded to these rotations of these patterns of neurons in this subspace coding mathematical representational space. So what that suggests is these brain waves are organizing neurons. These rotating waves of electrical activity across the surface of cortex are actually organizing the underlying neurons to make them do this murmuration, highly coordinated patterns. right? But here's the thing that really gets me about this is that, there's a direct mapping between movement in this mathematical construction of neural computation. This subspace coding here, this doesn't really correspond to anything, doesn't in principle correspond to anything meaningful in the geometry of the brain. This is a representation of how coordinated neurons are in this mathematical space where we reduce all the dimensions down to 3 dimensions. So this rotation here is in this mathematical representational space, yet there's a direct mapping between neuron coding in this mathematical space to a rotation on the surface of cortex. Now, there's no reason in principle why there should be this direct mapping, but what it suggests to me is that somehow computation is mapped across the surface of cortex. And the way the brain performs computation is moving traveling waves across the surface of cortex to sweep through this map of computation. And I think what this suggests is the possibility that your brain is an analog computer. Again, as I said at the beginning of my talk, is that the way we think about the brain, the traditional way of thinking about the brain, the classic way of thinking about the brain, it's a digital computer that uses connections to do computation. It uses ones and zeros, that's what those spikes and impulses are, and it uses connections to do computation, which is what AI is modeled after. Well, I think all this suggests is that your brain is not doing digital computation and connections like AI. Connections are important in the brain for storing information, but it's not how the brain does computation. Instead, your brain is an analog computer. So current models of brain function and AI rely on connectionism and digital computation, right? Well, analog computation is very different. It uses continuously physical quantities like waves to do computation. And you see an example here. Here's 2 sine waves. There's 2 pure sine waves. And you see as we slide the sine waves past one another, when they line up together, you summate to a sine wave twice as big. And when they slide past one another, they begin to cancel one another out until they flatline. So what we're seeing right here with just 2 sine waves is addition and subtraction. Not with the ones and zeros, but with these continuous variables between the ones and zeros, between the peaks and valleys. So your brain wave, this is just an example here, is just from one dimension, 2 sine waves. But your brain, these traveling waves in the brain are operating in 3 dimensions. And it's been shown that wave patterns in 3 dimensions can model and perform complex computations, including matrix algebra and differential equations. This is not theory. Analog computing has been around since 1400 BC. A slide rule is an analog computer. But importantly, Vannevar Bush from MIT, the guy who invented the modern way of funding science until recently, his first claim to fame was that he built an analog differential analyzer in 1927 that used wheels and disks to solve differential equations with 18 independent variables. They did that to compute missile trajectories for the Army, and it worked. Analog computing predates digital computing. In many ways, digital computing is kind of a bit of a sidetrack because analog computing is very powerful and it's energy efficient. In fact, analog computing is information rich. Instead of using just ones and zeros, and we think of the peaks and valleys of the sine waves as a 1 and a 0, analog computing uses all the values in between, not just ones and zeros. So it's information rich. It's also parallel. All the different variables in the equation are coded in different, everything you want to compute on is encoded in different parts of the wave. And the way you model an equation is you set up the geometry of how the waves are going to come together, how the traveling waves are going to interact. When they interact in different ways, you can model different equations. And then when you set up that interaction, you're modeling the equation. Then when the waves come together, because all the variables are there and the waves all there simultaneously, when the waves come together, they solve the equation in parallel. And that's very different from digital computing, where every equation has got to be computed one piece at a time. And as a result, analog systems are vastly more computationally and energy efficient than digital systems. I could put the word vastly 3 or 4 or 5 times there because the efficiency is remarkably different. Current AI, which is based on digital computing connections, is extremely power hungry. AI consumes enormous energy. The demands of AI are projected to exceed the global power supply. Yet AI isn't that smart. They're building nuclear power plants and hydroelectric plants to power AI, and AI ain't even that smart, right? Whereas contrast, biology does it differently. evolution favors energy efficiency. Your brain doesn't need a nuclear power plant to be as smart as it is. Your brain runs on the power that it powers to run a light bulb, 20 watts. Biology does not solve problems by throwing tons of energy at it. In fact, biology does the opposite. But a pretty good definition of life is local reversals in entropy with as little energy as possible. Okay, so biology, it's pretty clear that your brain is doing computation in a way that's fundamentally different from the way we think about it traditionally in the brain, or the way AI is solving the problem now. Namely, that your brain is organizing traveling waves that allow analog computation. In fact, these organized traveling waves are the raw materials for analog computation. Your brain is constantly generating exactly what it needs for analog computation. So I want to leave you with this hypothesis. Your cortex, maybe your whole brain, but especially your cortex. I'm sorry, I don't mean to say that connectionism isn't important or spiking isn't important. There's a lot of stuff your brain does that may rely on feed-forward, simple connectionism type computation. But real high-level computation may be done by analog computing. Your cortex functions like an analog computer. It self-organizes itself and computes using waves as are computationally tractable, and the waves provide highly energy and computationally efficient analog computing. So I want to offer the hypothesis that consciousness arises when the analog computation the brain is doing generates wave patterns, global wave patterns, and manages to unify the cortex into one organized whole based on the situation at hand, and gives you this unified experience of consciousness. And I'll leave you with that hypothesis, and also thank the students in my lab who are the ones who are doing all the work while I get to go around and tell people about it. Thank you very much. And thanks to our funders too. Thank you.
Earl, that was great. Thank you very much. I suspect you might have anticipated I'm going to ask you a question. I take the speaker's prerogative, but we'll have questions from others. From 2 centuries ago, Meyer and Overton showed that anesthetics all act on the same target, the unitary mechanism. And what you said before, that they act on different ones, I don't think is only partially true. For example, propofol does bind to GABA receptors, but also binds to microtubules. Ketamine binds to NMDA receptors, but also binds to microtubules. Dexmedetomidine, I don't think it's a true anesthetic. It's more like sleep. If you take a knife,
it is, yes, it is. But you can do surgery if I give you enough Dex.
But if you take a knife to somebody on Dex-Met alone, they'll wake up. So, and I'm not sure where it binds to alpha-2 receptors, but just take the other two. They bind to membrane receptors, but also microtubules. And there's compelling evidence from genomics, proteomics, optogenetics, pharmacodynamics, all that anesthesia acts on microtubules, and specifically only on quantum effects of microtubules, so it doesn't affect other functions of microtubules. So how would you reconcile that?
Stuart, you know what I'm going to say, because I've said it before, is that I'm open to the idea that microtubules play a role in consciousness and may even be the action of anesthetic. When I talked to my reductionist colleagues, I was referring to my reductionist colleagues, not you. But this is the way I was referring to the way traditionally people think about the brain. Now, as you know, we've talked about this, is that the microtubules act on the very high frequency in the gigahertz range, and that's hard for us to measure directly using our electrophysiological tools at that frequency range. But we are working with Demetrius Pronosis now to find a way to look for signatures of gigahertz kind of effects down in beats in the lower frequency. So I'm open to the possibility, Stuart, and in fact, I'm actively testing it. I'm just, I always let the data tell me where to go.
Glad to hear. One more comment, then I'll let everybody else jump in. But that is that we've actually been measuring megahertz. It's true that microtubules generate gigahertz, but it's much easier to detect megahertz from the scalp. And several studies have now shown that the triplets from megahertz go away under anesthesia, and actually under psychedelics increase frequency, intensity, and also split in from like 3 peaks to 5 peaks. So we're going to be pursuing that. And
that's just the kind of thing we're looking for, Stuart. Just the kind of thing we're looking for.
Great. Okay, we should talk about that separately. So thank you very much, and let's have now Tsuchuya over here. He's going to ask you a question.
Hello. About your data for the ketamine, What did you interview the patient who underwent this ketamine anesthesia, whether they had a dreaming or hallucinatory kind of experience during that dosage or not? And if they had an experience, but you also found the same kind of, phase shift, then doesn't it kind of raise a problem to your theory?
I so wish we could have done that, but our subjects were animals, not people. I can't interview them.
I see. But there are some data set with the ketamine, and then people report a dream and so on. So you would predict that then ketamine shouldn't affect this, disruption of the phase, right then?
We have found it does phases. This is an anesthetic doses of ketamine, high doses of ketamine. Yeah. Low doses of ketamine do something very different. They actually make your brain, they actually phase align the frequencies better than awake state. And they produce a more stable brain. And they also produce high frequency gamma oscillations. So unlike the other anesthetics, low dose ketamine is very different than high dose ketamine. When I talked about the phase shift, that was high dose ketamine, anesthetic doses. But low doses do kind of what you're suggesting. Low doses actually align the phases better. They actually make the brain more in sync with one another.
I see. Thank you.
Yeah, I agree with that. Over there?
Yeah.
Yes, thank you for a wonderful talk. I was wondering if you could comment on whether memories could be envisioned or could be explained or helped by the particular trajectories in phase space that you indicated.
I think we can understand how memories are formed by
not how they're formed, but let's say you're recalling. If you're recalling something like a chocolate sundae, would that correspond to a particular trajectory in this phase space?
It should, because this space is how the brain does computation. Doing that at individual level is very difficult because our brains are all individual.
I understand. I was wondering about your speculation on that. So.
My speculation is yes, it would. We should see that because that's how the brain does computation, by organizing these waves and moving neurons through this computational space. But the problem with individuals is the following: is that our brains are all wired a little bit differently.
Yes.
If you think about Eve Marder's work on the lobster pyloric motion, the pyloric motion is you move food down the intestine. Eve Marder Brandeis has shown that there's 3 ganglion in the lobster nervous system that do this, and you could wire these 3 ganglion together in 100,000 different ways and get the same pyloric motion. So it's not the individual lobsters are all different, but they're operating in the same principles. And our brains are the same way. Our brains are seeded with random connections, too many connections. And as you develop, you winnow away those connections by individual experience. So our brains are all wired up in a slightly different way. So the qualia, the hard problem of consciousness, the experience of consciousness, we could experience it all differently, but we'll never know. But they all operate on the same principles. And that's what my work is focusing on when it comes to the problem of consciousness.
Also, just one final quick question. Could these electric fields, if you had some sort of quantum behavior in the brain or quantum qubits of somehow in the neurons, let's say in the microtubules, could these brain waves be entangling them?
Sure, and what I want to see, and as I mentioned to Stuart, what I want to see evidence for is that this high frequency stuff has this downward influence down to the low frequencies, because we're seeing all this stuff in the lower frequencies. So if it depends on this action at the microtubule level, we should see this downward from the high frequencies influence down to low frequencies. And Stuart says he's finding evidence for that, and we're looking at it too. So stay tuned.
Over there, lady behind, second row.
Thank you for your brilliant talk. I would like to ask about specific interneuronal populations which you would think are crucial to the processes that you're studying. And if you could also briefly comment on what are the sort of developmental patterns that you see in the evolution of these oscillations. Thanks.
Yeah, I'm not a developmental. I don't study developmental neuroscience, and all these oscillations and traveling wave stuff are relatively new, so I don't have an answer to that. But as for interneurons, you touch on something very important. How do you get oscillations in the brain? There's 2 ways to get oscillations. One is excitatory neurons. They have these recurrent connections, both at the short scale and long scale through areas like the cortex through the thalamus, and those recurrent connections, like a snake eating its own tail, will naturally begin to produce oscillations. But another key component is the balance between excitatory action in the brain and inhibitory action in the brain. So the excitatory inhibitory neurons, the action between them is going to be very crucial. And there's more inhibitory neurons in your brain than there are excitatory neurons. So and mainly what we study when we study electrophysiology, we're studying the output, which is kind of the action of the excitatory neurons. And we need to also focus on the action of these inhibitory neurons. And we do have some evidence that certain brainwave frequencies, like the alpha beta I talked about, which is the control frequencies, that frequency specifically activates inhibitory neurons, which is exactly what you predict from our hypothesis that these alpha beta waves are acting as an inhibitory filtering mechanism in cortex.
Thank you so much.
Thank you.
Over there. We got a full house, by the way, here listening to you.
Oh, great. Hi, everybody.
Hi, Earl.
Hi.
Hang on. I have a specific question, but I just wanted to acknowledge beforehand just how well this model of lower frequency suppression, I guess, constraining or sculpting high frequency sensory input seems to map pretty well onto like a predictive coding model. At least that's the impression
Yes.
I get. Whereas the question I had was,
We have 2 papers on that showing the alpha beta acts as the alpha beta interactions that underlie predictive coding. We have some evidence, 2 papers showing evidence of that.
oh, legit, awesome. Yeah, I'll check those out. Okay, so I like the metaphor of gamma filling in like, or beta like the stencil and gamma like the spray paint. I'd wonder, are you familiar with Stephen Grossberg's filling in model?
Oh yeah, I know Steve very well.
Yeah, where you have like, just for the audience, you basically have boundary completion, which then gets filled in with, I guess, like color or brightness values. Wonder if you'd consider whether this model could describe something like that.
Certainly. I mean, I'll tip my hat to Steve Grossberg. And I said in the 20th century, we used to just focus on single neurons and no one focused on brainwaves because they were thought to be something you can only should measure if you can't measure anything else. Steve Grossberg he's the exception. There's always exceptions. Steve Grossberg was one of the first people to say that oscillatory dynamics play a big role in brain function. So credit to him for that. As for the specific details of theory, this is all what his theory doesn't talk about is traveling waves, and that's what we need to reconcile the two. But I'm sure they're reconcilable.
I think Grossberg's adaptive resonance will be consistent with the idea that EEG waves are actually interference beats of much faster ones. I actually asked him that when he was at our conference. So, and we have some preliminary evidence for that.
Oh, it's great.
Who else? Right there.
At what scale would a magnetic field disrupt the alignment and misalignment of brainwaves?
Well, we're seeing this across, the study where we found this is actually across a relatively limited amount of cortex. It's about 10 centimeters of cortex we're seeing this disruption. But what we've seen across several different studies is that after anesthesia, the front of the brain, frontal cortex, seems to get disconnected from sensory cortex. It's like the sensory cortex will still respond to sensory inputs, but they're no longer making it to the frontal cortex. And that's strange. What is disconnecting the two? And I think my hypothesis is that it's this misalignment of brain waves. Then the question is, what's the action of these anesthetics that can produce this disconnection in the front and back half of the brain? And some of my colleagues who I'm working with think it might be the different actions at different thalamic nuclei. That the sensory thalamic nuclei for the back of the brain have different set of connections and receptors and excitatory inhibitory balance than the thalamic nuclei that connect to the front of the brain, and that may produce a disconnection. But to my mind, it's probably going to result from this phase misalignment between the front and back half of the brain. But we're testing that right now with additional experiments.
Up here, somebody had a question? You had? Yeah.
Hi.
Hi.
Have you ever considered to do musicology studies to see how people respond to music and how this electrical field reacts?
I love that question. I haven't considered doing that, but I talk about this all the time. I'm a musician. You know, I say, like, is it a surprise that brains created music? Our brains run on music. I mean, like all these different frequencies I'm talking about, they all nest inside one another in a very harmonious fashion. When you see a 40 increase in 40 hertz oscillation, you also see an increase in 80 hertz oscillations, because that's an octave. So it's no surprise our brains created music, and we also know that that rhythmic input from the outside will entrain your cortex. So yeah, there's a lot to be studied there for sure.
Right there.
How would excuse me? How would your theory explain the difference between sleep and dreaming?
Hasn't yet. I haven't thought about that, but it hasn't. We haven't done the explanation for that yet. We have an explanation for how you get this slow wave sleep, whereas dreaming is more like your cortex goes into this awake state. It looks very much like awake state, but all these other things are inhibited, like motor output and is inhibited in your brain. So the prediction would be that the cortex would return to like the awake-like state. We would see the high frequency radio chatter during dreaming. And then how that you get into that state but still remain asleep is a subject of other work and other experiments. I don't have an explanation for that.
What do you think would trigger a dream from just sleep to dreaming? How would that happen?
Don't know. I mean, that's a great question. Sleep is a important mystery. We do know from work like Matt Wilson and MIT that during sleep, your brain replays experiences from the day in a highly compressed format and backwards and forwards, and that seems to play a role in memory consolidation, in sorting out the important stuff from the unimportant stuff. So that's what sleep and dreaming seems to do. How it gets to that state is, one of those questions to be answered.
Thank you.
Let me just say, Mate Blanco analyzed the logic of dreams, and it's more like a quantum logic than a classical logic. So dreaming may be just quantum information that doesn't collapse.
Stuart, send me that.
Who else? Right behind you.
Follow-up question. This one might be a little bit weirder, but I was curious whether you were familiar with the work of Stephen Lahar, who's one of Grossberg's students a long time ago. Mm-hmm. Oh, you know him? Oh, because he had a sort of very traveling way.
Wait, you talk about Stephen Lahar?
Yeah, Lahar.
I am very good friends with Stephen Lahar. He's a good friend of mine.
No way.
We talk about this stuff all the time. Oh, in fact, the buzzing your lips in the mouthpiece of a trumpet, I borrowed that from Steve Lahar.
That's great, because I just hung out with him in Boston. That was his metaphor. That's interesting.
Oh, yeah, you recognized it.
Yeah. So, I mean, question is moot.
I'm sorry
He does great cartoons, doesn't he? Isn't that the guy who draws great cartoons?
Yeah,
neuroscience cartoons.
That's him.
I've stolen a number of them from him.
No way.
Go ahead, please.
We're all stealing from Stephen Lahar.
Oh, that's amazing. Okay, well, I was going to ask if you're familiar with his, I guess, like, he uses the term non-linear optics, but very sort of traveling wave.
Clifford algebra and how oscillations are great for representing infinity because you get decreasing Weber fractions and you get compression of scale. Yeah, Steve and I have talked about this over many a beer.
Oh, fantastic! He does like a beer. I know that much.
Yeah.
His flame front thing seems like the resolution to the Grossberg traveling wave thing.
Yeah, he'll be thrilled his name came up at this meeting. I'll be sure to tell him.
Fantastic. Okay.
Other questions? Over there. To your left, yeah.
Yes.
Hi, Earl. Thanks for your description of the analog processing. I'm taking from that you run your bass through a tube amp. But that's not my question.
I love the sound of a tube amp, but they're inconvenient, so I actually have an analog circuit pedal that emulates the sound of a tube amp.
Excellent.
So I'm cheating a bit, but still analog.
Excellent. I think there's a question of coordination. You mentioned that the waves across the cortex, outside the cortices, are fed or at least emergent from the activities of the neurons below. And at the same time, they are influencing those and substrates. So it seems like, how does that coordination work? Where's the information and the input for that?
Well, if I had the answer to that, we'd have the brain all figured out. That's the million-dollar question right there, right? I mean, how does this organization come about? It must be some form of representation, because that's what the brain does. It represents information. So if I look at a clock on the wall, there'll be a representation in my visual cortex of the clock on the wall. Well, the way I think of executive, this top-down control, this organization, is that in the front of the brain, there's a representation of your goal and what to do right now in the situation relative to that goal. Just like a representation of a clock on the wall, there's a representation of this goal and means in your frontal cortex. And then the frontal cortex sends this pattern information back to the rest of your brain and says, go do this. So in the end, it's going to be an information representation problem. But interestingly, analog computing kind of solves this whole problem in a way, because if what it says is that your current brain state, your current thought is this pattern of excitation and inhibition in your cortex determined by these wave patterns, right? Then the wave patterns come together and by computational principles, they produce another wave pattern. And that's what thought is. Thought's constantly changing. Our waves coming together and in a principled way producing new wave patterns. And if that's all there is to, if that's what there is to thought, then you no longer need homunculus, you no longer need a little person in the brain decoding information. And neuroscientists still talk about these, they say, we ran a decoder on this. There's this assumption that somehow downstream neurons are decoding the information from the neurons you're studying. Well, what's decoding? What's the decoder in the brain? There is no decoder. It's just waves begatting waves in a computationally principled way. So brain states shift to different brain states using this analog computing. Bam! You'll no longer need a decoder. You no longer need a homunculus. How this all comes about in a mechanistic detail way, I'd like to know.
Okay, thank you.
Sure.
Any others? Where? Where? I don't see. Okay. Oh, okay.
Thanks for a really great presentation. I'm a newcomer here to all of this here at the conference. My name is Radar. I'm from Aalto University in Finland. And what you said really resonated with me. And actually, as it happens, the previous question was an excellent bridge to my question. Because
sorry, was resonate, was that a pun?
It was a pun. And I actually intended it.
Good.
Anyhow, so what we're working on is to refurbish the usage of the word meme at Aalto University, whereby we would then have memes, meta-memes and hyper-memes as strictly formalized sets of representations. So my question simply to you is whether you have encountered any other formalized, rigorously mathematically set theoretical formalism or formalized framework for actually taking these patterns at different levels. I mean, we could call them something else, and I received all kinds of comments, oh, it's memes and memetics. However, as we have found out in our extensive work, it works quite well as a formalism, as a set of representations, as you just happened to say. So have you encountered other efforts or rigorously formalized
no, we haven't. I mean, I'm hearing from you, so more power to you. I hope you keep me up to date on your work. Sounds very interesting.
Yeah. I put up a first paper on OSF after my poster presentation.
Oh, fantastic.
So I'll share that paper with you. Thank you very much.
Please do, yeah.
Any other questions? One more in the back, and then we got to move on. Earl, thanks for your time. We really appreciate it. One more question.
My pleasure.
I have 10 more, but I'm going to keep them inside.
Oh, we'll talk, Stuart. We always do.
Does your account for how, like, the mechanism of action of anesthetics in terms of, like, disrupting with creating these, like, phase, out of phase, low frequency oscillations, maybe suggests that there's actually maybe a fragmentation of consciousness more, and that there are like islands of awareness during the experience?
That is possible. I mean, when these animals, when they recover from anesthesia, we see the brain flip into awake state, then back, they often go back and forth as they're recovering to consciousness and unconsciousness. And we see this flipping back and forth between the two different states of low frequencies and high frequencies. Whether those low frequency situations are isolated bits of consciousness in different parts of your cortex, yeah, it's possible, but that would be a very different form of consciousness than normal awake consciousness.
Like an
it's
octopus.
possible, but it'd be very different.
All right, thank you.
Thank you.
Okay, thank you so much. Let's give Earl K Miller a round of applause.
Thank you. Thank you all.
Thank you, Earl. Okay. Our next speaker is Matthew Larkum, and he's, I forget the exact title of his talk, but while Matthew's getting ready, let me say about one announcement. For the party tomorrow, we're going to have the poetry slam, and we're going to have beer tomorrow, and food. And during the poster time, there won't be posters. So after the concurrence, we'll go to the third floor and have a party, including, as I said, beer and a poetry slam. If you want to present a poem, we'll post one of those QRS things tomorrow that you can send it in, so we have an idea of how many. And our next speaker is Matthew Larkum, Dendritic Decoupling Hypothesis of Anesthesia, Dendritic Integration Theory from the Humboldt University of Berlin. Matthew, welcome.
Oops. Excellent. Well, it's an honor to follow two talks such as those, and hopefully the resonances continue. I'll talk about a lot of things that are, I think, completely compatible with what Earl was just talking about. And it's also one of the 300 theories that Robert was talking about right at the beginning. And it's also a very bottom-up theory of consciousness that we've been looking at now over several decades. But in the end, I hope you'll agree with me by the end that it converges on a multi-level set of claims. I actually gave a talk about this about 2 or 3 years ago, and I unfortunately couldn't come to Tucson because that was in the middle of COVID, so it's nice to get an opportunity to give this in person. And so I'll just quickly go over the claim here. It's based on an observation, essentially, that the cortex is made up of 70 to 80% pyramidal neurons, and the pyramidal neuron itself is a very special beast. It's clearly the evolution has clearly decided that this is the cell type of choice for a complex computational device like the cortex. And when you look closely at both the morphology and the properties of these neurons, it becomes clear that they basically have two particular regions where they collect their inputs, but the bottom and the top, or the basal and the apical compartments. And what we found in studies now going back 25 years, that the properties of the cells are also aligned with these, with the architecture or the anatomy of the cortex, such that to a first approximation, the external stream of information is coming to the bottom of this neuron, and the internal stream, that's whatever is generated by the cortex itself and other regions that are essentially giving feedback to the cortex, come to the top of these neurons. And what we found at the physiological level was that there are channels in the apical dendrite that lead to a really explosive event called a calcium spike. And this actually leads to the detection at the cellular level of the integration of these two streams. So in that sense, every neuron in your cortex is doing a kind of prediction. At the network level, this then amounts to since all of the different regions of your cortex are performing some sort of modular or low level or lower level operation, such that in sensory cortex there are low level features, in then higher cortical regions there are no longer features in that sense, but there are nevertheless tasks that have been computed at higher and higher levels. And the idea would be that if you're perceiving, for instance, some object that, as we know anyway, that these features would be broken down or processed in particular regions of the cortex, which would get in the external stream input to the bottom of the neurons, and in the internal stream, this would come via the horizontally running fibers in layer 1 to the top of these neurons. And what we predict and now have been able to show is that this ignites a certain subpopulation of these neurons that becomes an ensemble that, without some homunculus, then signal in their really profoundly different firing that these neurons are, let's say, matching the external or the internal prediction of what it is that is being perceived. And I suppose the attractive thing about this as a theory is that it operates then at multiple levels. We have intracellular events that we predict. There are outputs from the neuron that we would predict. There are network level features of this as a theory, and in principle this should work at the cognitive level, and indeed, so far as we've been able to test that, this is the case. So the last thing that's really appealing, I think, about this as a theory is that it gives us things that we can actually measure at the cellular level that should correspond over all of these levels. And in particular, we can do 2-photon imaging in live animals. And because this event that we're looking for is due to calcium flowing into the apical compartment of the neuron, that means we can use all the latest technology in recording calcium events with 2-photon imaging. And indeed, we've been able to see these events going off that match, for instance, the moment of perception in a threshold detection task, and so on. And so this is ongoing work, but it looks very promising at the moment. And it's described in a few papers now, so I can recommend those for you. The time at which we thought we might be on to something that we could claim links to consciousness comes from a study that then I talked about a couple of years ago for the Tucson version of this conference, and it really is it was due to a chance finding about the loss of consciousness under anesthesia. So we were looking at what happens to these neurons, to populations of these neurons when the dendrites are activated. And to do this, we used an optogenetic technique in mice where we put channel rhodopsin in these pyramidal neurons, and using a mirror, or a sort of fiber optic way and a mirror to give a horizontal beam of light, we could activate the apical dendrites of these neurons and ask the question, what happens now? Do they go into the mode that was predicted from many years of recording from these neurons? And sure enough, in the awake state, one sees the evidence of bursting from these neurons. I won't go too deep into the electrophysiology here, but this was a robust finding and quite expected. What surprised us at the time, and I don't think was as expected, is that under anesthesia, this flatlines. You might think that this is to be expected because the anesthesia is obviously dampening networks and causing neurons not to fire. But actually this is really, really astonishing, because the neurons are expressing channel rhodopsin, and so the blue light on the dendrites is causing current influx into these neurons regardless of what's going on at the network level. So this tells us that there's a cellular mechanism that's blocked by the anesthetic that prevents the top of the neuron signaling to the bottom of the neuron. And So we can see this decoupling, and the hypothesis, since these cells are ubiquitous across the whole cortex, and is one of the remarkable things about the cortex actually, that the architecture is roughly the same no matter where you look in the cortex, from the lowest sensory regions to the higher, to the top of the hierarchy in the frontal cortex. You have the same neurons doing apparently the same algorithm, if you like, for computation. And of course, if this, and we were subsequently able to show that this mechanism is operating in different brain regions, from primary visual cortex, primary somatosensory cortex, right up to frontal cortex in rats. And so the idea is that, oops, I'm going backwards, that this is therefore a ubiquitous decoupling of the feedback stream that comes through the top of these neurons that we already suspected were really important for conscious perception. And lastly, we were able to show there's an influence of the thalamus on this. So what I'm showing you here is a diagram, or actually data from, this is a higher order thalamic region in mice, and their projection to the cortex. And you can see two thin lines basically. So the first order projection from the thalamus goes to the layer 4, and that's often what's focused on in textbooks. So there's a higher order connection from actually multiple regions of the cortex that has a profile that's more like this. Sometimes it's more layer 1 than what is here, layer 5A. Sometimes it's more layer 5A. But here in this particular region, which is the somatosensory region of a mouse, you can see it really clearly in both places. And actually, there's not much theorizing about what this middle region does. In any case, this is right in what you might call the coupling zone of the pyramidal neurons for linking the two compartments of the neuron. So it was an obvious experiment to block the thalamus and ask what happens. So we blocked this region of the thalamus, and we basically repeated this experiment in vivo. So here we're expressing channelrhodopsin in these neurons, putting light on the end, and then controlling this under a normal awake animal. And now without anesthesia, we can dampen the effect of the light on the apical compartment at the bottom of the neurons. I should have actually added that the axon comes out at the bottom. So whatever happens at the bottom is what gets transmitted to all the other neurons. And blocking this action essentially cuts off the influence of the top of the neuron in the output of this neuron. And this wasn't due to any action on the apical compartment, but as predicted, because the channelrhodopsin's in the apical compartment, should also not be affected by anything other than the turning on of the light. So this included or means that this thalamocortical connection is also maintaining the coupling across these compartments. So we've got these results. This was this is an extracellular way to look at the population response, both in the control, the red line here, and under anesthesia. We actually also showed that various neuromodulators can affect this coupling too. Just to say, it's a really complex story. This seems to be a bottleneck of effects, if you like, across these really important neurons, such that under various different conditions, the top of the neuron can suddenly have a no or a variable influence on the cell body. And to a first approximation, this gives then a way of understanding a possible mechanism for anesthesia, which is due to, we're claiming, some weak link or a sensitive link in the apical basal axis of these neurons. And essentially what that means is that if this link gets blocked, you block or if you like, if this one mechanism that is ubiquitous across the whole cortex gets regulated, then you will block this loop or that loop, and the whole chain of causality will be broken, such that you will lose what's in essence then a very integrated and interconnected system. So in the meantime, so since I showed this some years ago, we've been working with a team at Sydney University in Australia who are looking at this from let's say a whole brain modelling perspective, trying to incorporate the different kinds of thalamic connections and their laminar targeting in the cortex with a model of pyramidal neurons, or neurons, let's say, that have two compartments instead of just the one compartment that's usually used, and they've built in a special sensitivity such that the top can, under certain circumstances, be influencing the bottom and not under others. And here they're paying particular attention to this. One word that's used often for describing this higher order thalamic connection is a matrix connection to the cortex. It's a less specific connection that influences the top, and as we saw, the middle of the cortex, versus the sometimes labeled core connectivity from the first order thalamic regions that tends to go right to the middle of the cortex and to layer 4 and propagate through there. And in this way of constructing the model, you therefore have situations where the influence is more core, and other situations where the influence is more matrix, and they can play around with this, and then they can ask questions: what happens under anesthesia and so on, and look for various properties, some of which we've just been talking about in the previous talk, that should correspond to the addition of the anesthetic, and pops out of the model just by having two compartments and playing around with this connectivity. So I won't dwell on that, only to say that it's quite actually beautiful to watch how the model very simply captures this property, if you like, of what anesthesia would do to the whole system. And finally, in the modeling sense, it's one way to construe what's going on in the cortex and how this relates to your perception, is that you can think of a cortical column in this sense as outputting a computation, if I could put it that way, which is basically the probability of that given feature given the context, where the feature-specific information is dominating or driving the baseline firing of this cell, but context-specific information, and that means everything else except this feature, everything else that's being given by the brain to the top of the column, that's being computed via this huge nonlinearity within the cell itself, such that what you get out of this is a approximation to the probability of that actual feature that's coming from the outside world, given what you know and what you believe about how that integrates with your quote-unquote model of the world. So this would be nothing other than the way you model the world would be what happens as a computation in that cortical column. And you have to imagine that this is being done massively parallel way across the whole cortex at multiple levels, from very low level features to very high level features. And so the sum total of all of this is the probability of the interconnection of all of these features at every level, given what you know and what you would predict over that space. And what we're looking at here is, in terms of modulating that and grabbing hold of the context part of this computation, there's essentially a highway between these two regions of the cell that can be adjusted, it can be manipulated. So in the meantime, we've been able to look at this effect in vitro, and we were just starting some of these experiments last time, and we were able to show you can repeat the whole experiment in vitro by expressing channel rhodopsin in neurons, slicing up the brain, and then doing the whole thing in the dish while you patch the cell bodies of these neurons. And then, of course, we do these experiments upside down because it's in some sense anesthetized, or at least it's not anesthetized, but it's not awake at the same time. So they're decoupled in the first instance, and we can recouple them by putting on agonists for the metabotropic modulatory neuromodulators that were blocking this coupling in vivo. So if we go the other way, we can recouple neurons in vitro. And we've been looking at that now more closely, and we're still not finished, but we see synergistic effects of two neuromodulators that we were showing, each of which were working in vitro. I don't have time to go through all of this, and we're not finished with this, unfortunately. But it's very promising. We can reproduce these effects in the dish in terms of coupling between these neurons. And the biggest effects we're seeing are the ones that are most proximal. So we think it's going to end up being that connection from the thalamus that comes in layer 5A that is really serving the coupling between the two regions. We are also just starting to look at the effect of disturbing the microtubules in the apical dendrites of these cells. I really wish we had this finished before I came, but it is what it is. I fought hard to try to get people on board with this. I finally got a really keen student who's in the middle of this project now. Of course, amongst the many things that could be said about microtubules, they're also very much important for the stabilization, for the linking of the top to the bottom of the cell, and so they also putatively could affect the transmission of signals from the top to the bottom of the cell. So it looks very promising at the moment. We don't have a lot of data. This particular drug, which disturbs the microtubules, affected the coupling from the top to the bottom. We see this in a few different drugs. There are lots of issues that I'd be happy to talk about that are coming up in terms of differentiating the effect per se of these drugs on this excitability of the cell from the effects on the coupling. And we're in the middle of that at the moment, but it looks very promising. There's even reverse effects for drugs like Taxol that tend to stabilize. This is a recording at the cell body of a neuron with patch clamps. So this is membrane potential. So you're looking at I should have taken some time. It's very preliminary data. This is an action potential that's being generated by the light from the light being applied to the dendrite before in black and after the vinblastine. And the vinblastine here is being applied intracellularly. And that's another problem we've got here is trying to get these effects at such that we can say that it's really on this neuron and not at the network level and so on. As I say, this is a work in progress, but it looks very promising at this point. I guess that the devil's going to be in the details for what we end up being able to say exactly what's going on in the meantime. So we find ourselves in terms of what we're calling dendritic integration theory, which I don't know if I'm happy or sad to say is just one of 300 theories that Robert was talking about, is that it's proven to be very predictive for I think understanding what the cortex is doing, but also for generating new experiments. And in fact, I think it's such a low-level claim about cortex that one should expect it to be congruent with many other studies and many other claims about consciousness. And indeed, I think if we look closer at everything that Earl was just saying, you get lots of overlapping claims here. There's a couple of things that have happened in the meantime that have been really salient. One has been we've been able to make claims about memory which I'm not going to talk about today, which is why that cross is there, just because there's no time. But one thing I thought would be interesting in a session that's talking about attention and oscillations and so on is a claim that comes out of a prediction that came from what at first glance would seem like a bizarre link to this theory. So I had a postdoc come to me almost out of the blue one day and show me something that was not quite, wasn't this particular picture, it's a published paper, but another picture looked very similar to this. And what he was showing me and was asking me about was this layer of cells at the bottom of the cortex, which is labeled layer 6B, or sometimes layer 7. Cahal called it layer 7, for instance. It's been known about for over a century, but nobody really talks about it. It's been very understudied, and it's very difficult to imagine what these cells are doing. One thing I did know about this set of cells, and knew since we started working on the pyramidal neurons, was that these neurons connect to layer 1. And in dendritic integration theory, anything that goes to layer 1 is really significant, because that's essentially context coming into these neurons. So I said to this really gifted postdoc, Tim Zolnick, in the meantime now has a couple of really excellent publications on what these cells are doing. Go study that, because that's going to end up being really interesting. It exceeded all my wildest dreams about how interesting this would be. One thing to say, what you're looking at here in the background that I'm sort of covering up now is from another paper where they're showing the expression of 5-HT2A receptors. So these cells at the bottom of the cortex, as well as the major pyramidal cells, express the receptors that are most linked to psychedelic action, although that's probably simplifying it too much. But never mind, they express 5-HT2A receptors extremely. They also express basically every neuromodulatory receptor you've probably ever heard of. They're quite famous for expressing orexin, which is a really important in the sleep-wake cycle. So the orexin excites these neurons, and you need essentially orexin in the system to be awake. They express cholinergic receptors, dopaminergic receptors, and a host of others that I won't get into. So I was just alluding to the fact that these neurons project to layer 1. When Tim looked at this more closely, what he found was that they don't just go to layer 1, they also go to layer 5A. And at that point, it started to become, I think, really intriguing. So if we look at the projection of the layer 6B to the cortex in terms of which layers they're influencing, and you compare that to what this higher order thalamic region does. So this is a particular higher order thalamic region, the posterior medial nucleus in a mouse, but basically is emblematic of this matrix connectivity that I was talking about before. They go to exactly the same layers of the cortex, and it's not just the same layers of the cortex, but the same sublayers of this cortex. So they both go to layer 1A, the very top of the canopy of these trees, and they both go to layer 5A, which is the upper part of layer 5, which is exactly in the coupling zone. So if you imagine this now in this scheme in terms of all of the neurons of the cortex, you've basically got this thin layer at the bottom of the cortex regulating exactly the two regions of these neurons to maintain, let's say, the connectivity of the top to the bottom. So in one of these studies, Tim was able to show that there's another property about layer 6B that's really interesting that I didn't know when I sent him off to look at this, which was that they only receive cortical inputs. So essentially, layer 6B is the only layer of the cortex that receives no, or to a first approximation, no thalamic input. So it's got its own, it's shut off, if you like, from the external world, at least from the primary drive. It, on the other hand, it's got the projection that we were just talking about. So in some sense, the layer 6B could be thought of as the cortex's second thalamus. This is an internal thalamus that's able to control, if you like, the same loop that's going on between the thalamus. And I guess that we're not the only theory of cortex that would posit that there's something really intrinsically important about the thalamocortical loop and to consciousness. I think these low-level theories of consciousness get some criticism, which I accept, which is that they're not really an explanation. I can imagine if you're comparing this with some of the higher order theories or some of the theories that really ask this question directly and up front. In the end, I can't tell you why the thalamocortical axis is so important, but if you take that for granted, then you've got to include 6B in your worldview as to what effects it would be controlling, because it's not just controlling the node or the two nodes, the pyramidal neuron node and the thalamus, because I didn't get to say that yet, that it has one other connection which is down to the higher order thalamus, but it's controlling the two nodes of the pyramidal neurons that project to the higher order thalamus. So it's basically got the thalamocortical loop by the scruff of the neck, and is in somehow controlling all of the nodes of this output. So apologizing now for the detailed electrophysiology, but here you're seeing an optogenetic experiment where light is being shone on the 6B channel rhodopsin expressing neurons. That means that we're asking the question, what does the output of these 6B neurons do to the pyramidal neurons? And we're doing this experiment in vitro. And we can put the light wherever you like, so we can ask what's the input from the 6B to the top of the pyramidal neurons, what's the input to the middle of the neurons, and we can play God now with the 6B connection and look at what it does to the neurons. And essentially what you're looking at are 10 pulses in a row, seen from the perspective of an electrical recording at the cell body, and you can see it's going berserk. I hope you could. Well, actually, it's cut off, so you're not even seeing all the action potentials. You're seeing essentially the underlying drive. What's going on here is it's setting off what are the equivalent of electrical bombs in the apical dendrites of these neurons. And it's doing it, we've found now, through NMDA receptors, because this drug AP5 that blocks NMDA receptors completely shuts it down. What you get left after you block the NMDA receptors is the AMPA component, and without getting too technical, that's the bog standard receptor type the glutamate receptor that would be what most people postulate as the typical driving force of neurons. And if you've done a course on neuroscience, you will have heard of the NMDA receptor being involved in plasticity. But there's another thing going on with NMDA receptors, because they can basically cooperate, and that's the sense in which it becomes a bomb in the apical dendrites. And if you get that right, or if the brain gets that right, then you get orders of magnitude more bang for your buck from the NMDA receptors than you do from the AMPA receptors. And this is a case in point. So the 6B connection to the top of these neurons makes them go berserk in a way that if it was just AMPA, the ones you know and love, basically have no effect from this particular connection. We can do the same operation with the thalamic input. This higher order thalamus also has a strong effect on the distal dendrites, not quite as strong as layer 6b, but quite strong. And again, blocking NMDA receptors blocks those. Just because for completeness, we did this in the 2 types of pyramidal neurons, but because that's, I think, too technical for now, I'll skip over it. The last thing to say about this, and this will drive now to why I think this might be really important for attention and selection, if you like, of neuronal activity, is that these connections are facilitating. By that I mean that. So if you take the thalamic connection to the middle region, now you can see the light has changed. The light is going on to the proximal region, not to the top of this neuron. What you get for the thalamic connections is what's called a depressing synapse. So that every time you give a light pulse here to ask the question, what does the thalamus do to the middle of, essentially to this coupling region, it gets less for every pulse that you give. If you do the same thing with the layer 6b connection, it becomes facilitating. So this seems like a detail in the back that you probably don't want to care about, but I think it's really important. What it tells you is that the thalamocortical connection has a tendency to die, if you like. It will not sustain itself because the more that you do it, the less effective it gets. In contrast to the 6b connection, which is getting bigger and bigger the more that you use it. And not only is it getting bigger and bigger over a train, but each subsequent train you do gets bigger. So here what you're looking at, if the dark blue line here is the same thing done with a pause and then a repeat of something like 10 seconds. And you can see that that's by trial 5 and by trial 10, which is, you should focus on the purple lines. They're getting bigger and bigger, so they're facilitating within the train, and then they're kind of, this is a second order facilitation that's going on with a break, which means that, if you like, the cells are remembering the fact that they were excited via this connection, and this is the connection, bear in mind, that would be maintaining your conscious awareness of whatever on whatever feature or cortical column is in your memory. So we are hypothesizing that in some sense 6b is a conductor of your attention. And when we say this, we're not claiming this is the theory of attention, but this will be an important factor. This will be essentially a subliminal factor in your attention, because presumably it's the thalamocortical loop that is what you're quote-unquote aware of. And so you're actually not aware of the 6b. So this will be something more like a gist that is feeding into the things that you can become aware of, and so on. And without going too far into this, I just say that this is now, this I think fits beautifully into a number of different claims about thalamocortical loops, not least of which we were just hearing from Earl Miller that would be important. I should, if it's not immediately clear either, just to get another resonance to the previous talk, the EEG that one records from the scalp, that is really the basis of the oscillations that Earl was talking about. These are almost entirely, or let's say completely dominated by the flow of current around the axis of the pyramidal neurons in the brain. So the dipole that is caused around the pyramidal neurons is almost entirely what you're recording when you record an EEG. So it makes perfect sense that what one sees at the single cell level here will manifest in terms of the kinds of frequencies or oscillations that you see. One of the ways of testing this was to take an animal that was already sleep deprived and therefore going in and out of slow wave sleep, and we could do this cool experiment. And I want to emphasize that all of these experiments are coming from the theory where we say, okay, then we should predict this next experiment. We'd never have thought to have done this experiment otherwise. Put channel rhodopsin in these layer 6b neurons, and now we record in the depth of the cortex. We put light on the brain and suddenly excite the axons of the layer 6B neurons and essentially ask what's going to happen. And just to decode this, the first activity you're seeing here is the slow wave sleep in a kind of delta rhythm that this mouse is experiencing. And when the light goes on, you're just seeing it pulse here every now and again. You can see the light go on and you'll see the whiskers move and something happens. At the electrical level, in this cortical column, it goes straight into high gamma. And this bottom trace here is the expansion of this box. And so you'll see it going from this delta current straight to high gamma, which is very much associated with attentive states. And quite remarkably, after the cessation of the light, it goes straight back to high delta, to delta, I should say. And this is quite unexpected also, because if it was really quote unquote waking up, it would now go through a series of steps to get back to delta. Whereas clearly this is like a switch. The 6B operation on this circuit is to put it straight into the quote-unquote attentive state. We don't know yet, and this is now going to have to be the next few years of research, whether the animal is, in fact, attending to something or whether it's still asleep and so on and so forth. So there's some interesting possibilities there. But anyway, I'll skip over this and I'll try to come to a close now, but basically where we are now is wondering to ourselves what 6B is doing in terms of the coordination of activity in the column. And of course it could be causing kind of coherent activity of all of the neurons, but it could also be playing conductor and turning on the neurons in some special way, depending on the activity that we are yet to discover that's going on in 6B. It could also, of course, be finding ensembles of neurons and coordinating those. And this could also be happening at the level of the whole brain. So you can imagine that was the look within a column. You could also imagine that since these columns are interconnected like this, that the same set of questions can be asked. Is it coordinating the let's say coincident firing of neurons around the brain, or is it coordinating which column should be now under your attention, or is it trying to select for ensembles of neurons around the cortex? So to summarize, what we're basically claiming, and this is coming, I think it doesn't make any sense to talk about 6B without talking about dendritic integration theory as a whole. We're claiming that this 6B, which is right at the bottom of the cortex, is impinging on a really important loop that controls your consciousness, and that this is basically another ubiquitous feature of the cortex that you find all around. If you put this into these higher models, you can quite quickly see that this is another collaboration with the Shine Lab, Max Shine in Sydney University. There's several ways now that they've been able to show that controlling this loop essentially pulls out or coordinates, selects the different loops of the cortex. And so I think what we have to do now is imagine that there's another really interesting thing that's not just deep in the cortex, but probably subliminal to cognition, that is doing the job of the pre-job of what eventually becomes your conscious perception. And it's got all the neuromodulatory receptors like 5-HT2A and cholinergic receptors and so on. And so this is an extremely complex set of possibilities that we're just now doing the first experiments with. All right, and with that, I'd just like to thank the people who were majorly involved in this. And Modotaka Suzuki, who's since left the lab and has now got his own lab in Japan. and Tim Zolnick, who did all the 6B stuff, and a host of other people that have been behind several of the experiments. Thanks for your attention.
Thank you, thank you. Questions? Over there. Can you run?
Thank you. Thank you for your presentation. In terms of layer 6B and its peculiarities, even in the perinatal stages and embryonic stages, you can see that it is a remarkable region. These cells are the first to assume their corticostriatal positions. We also see, we've been looking at interneurons there, and they seem to be far further into their developmental trajectories and mature in their morphophysiology already in the perinatal stages. Now, my question is actually to build upon your speculations of the function of the layer 6B. In the adult brain, you see it as a region very crucial to attention. But then in the embryonic stages and in the perinatal stages, what would you speculate is the role of that layer, considering that it seems to be already quite mature?
Yeah, that's a good question. So yes, that's right. It's one of the earliest developing regions. It's quite, it's not exactly homologous, but it's quite overlapping with the claustrum, in fact, which is also in the subplate. And one of the features one of the things I didn't get a chance to say is that although it connects to layer 1, it misses all of the tufts, the apical dendrites of the layer 2-3 neurons there. And I guess that's a developmental aspect of this, that presumably it wires up to the tufts of the layer 5 pyramidal neurons during this initial stage when there is no layer 2-3, and then as the cortex expands and inserts, if you like, it must have some shutdown process for forming connections. And this is a really remarkable thing in itself. So despite the fact that you've got really complex dendritic trees that are all covering each other and that are presumably really important for cognition, it misses all of those. And my guess is that it's very important in these early stages for ramping up, let's say, the predictive idea. I'm guessing, although this is really speculative, that you need those early moments to ground the system in the external world. And this would be all before you generate too much predictions, so when you don't have predictions essentially in experiences, and that from a cognitive point of view, you want to be maximally grounded to the external world. And this would be a way, if you like, of forcing that issue right at the beginning and making sure that the tops of the neurons are let's say, well embedded in a world that, quote unquote, exists, where the outside world would be very influential before you start generating your own internal model, as it were.
Thank you.
Okay, let's go. Is that Par in the back? And then we'll go to now.
Thanks for a great talk. I have an easy question. Those cells in layer 6B, is the effect of 5-HT2 agonism excitatory or inhibitory? Do we know? And a second question, which is more complicated perhaps, what is the relationship between the decoupling, the dendritic decoupling, and corticothalamic oscillations that we see during sleep and anesthesia? Are they connected? And in that case, is one causing the other, or is it more of a chicken and egg situation?
Okay, so the first one is that it's excitatory, and so we've been testing psychedelics and so different psychedelics, LSD, psilocybin, and a few others on their effects both on the 6B and the layer 5 neurons. To our surprise, it seems that they have a depressing effect on the layer 5 activity, but an excitatory effect on the 6Bs. So I'm not quite sure what to expect in the final analysis at the systems level. Okay. The other question is, what do we expect on thalamocortical rhythms? I think, in principle, that's the experiment that we tested, or at least that was the rationale behind the experiment that we tested, was that what would 6B be doing to the what has generally been seen to be thalamocortical rhythms, and it was completely dominant. So we think that whereas the layer 5 cells have often been attributed, if you like, to controlling in the final analysis some of the important oscillatory states, we think 6B is controlling the layer 5 neuron, and therefore is even from a causality point of view is even more important in terms of dominating the system. What we don't know yet is when and how layer 6B is operating. So we don't know when and how it's impinging on what the layer 5, the deep layer 5 neurons are doing for this important oscillatory loop. But it is causal, no question.
But wouldn't you need thalamus to kind of synchronize these oscillations on a larger scale?
Yeah, so there's a lot that's missing in that simple description and that simple experiment where you just excite the 6B. So there's also, of course, the connection to the reticular thalamic nucleus, which is doing an important inhibition of the other thalamocortical regions, and presumably has an important aspect of timing and let's say the creation of the various thalamic rhythms. The 6B has only connections to the layer 5 neurons and 6A interneurons. So it's doing some important operation with the inhibition of the deep layer, the deepest layer of the cortex, layer 6A, and those inhibitory neurons will be really affecting the layer 6 pyramidal neurons, which are projecting to the thalamic reticular nucleus, and therefore affecting these rhythms. So in the final analysis, yes, it's going to be more complex than just what we saw from the effect of 6B. On the other hand, I don't know of anything that's quite as dominating as this connection. So I'm going to guess that it's with and without 6B will be completely different stories.
Thanks.
Okay, last question up here.
Hey, so I was very curious about your experiment on the ongoing preliminary experiment on the microtubule interference.
Mm-hmm.
But I'm really curious about what you would expect about the action of that particular drug, Blanstine or something like that?
Vinblastine.
Yeah, Vinblastine. Yeah. Because if you interfere with the microtubule, then consequently you also probably change the configuration of the surface expressed ion channels, right? Probably.
Yeah.
So if that's the case, then disruption of the microtubule affecting on like expression of the ion channel on the surface, removing it from surface into the cell membrane, that would probably already explain the failure of the propagation from the dendrites into the soma.
Yeah,
so what you would like is really interfering some kind of Stewart or Rogers prediction about aspects of the dendritic computation and the microtubule related phenomena without affecting on the surface ion channels.
Yes.
Would it be possible?
Well, so this is exactly where we're getting stuck at the moment, and we really need to.
I can answer. Can I answer that?
Yeah, sure.
So 30 years ago, when we first proposed this, people went to the squid giant axon and squeezed all the exoplasm out of the axon, and filled it with saline, leaving only the membrane with the ion channels, and they still conducted perfectly well. So on the one hand, we're criticized because, well, how could the microtubules affect the ion channels? And now you're saying just the opposite. But spikes may be completely different from what's happening in the dendrites. I think that's true. But if you get rid of the microtubules in the axon, you still get a perfectly good spike.
Well, in the apical axis, we did see effects on the excitability of the cell. So right at the moment, we've got a patch electrode on the cell, and we can just record the passive properties of the neuron. And so we control for that in each of these experiments. So the reason why I still am optimistic about this effect is that we see a larger effect on the coupling than we see on the electrical properties of the cell. But we can see effects on the properties. Well, so if we inject some current at the cell, we can look at the response of the cell to that current injection, and this we call usually the passive properties of the cell. So the input resistance and the threshold for the firing, the various the time constant and so on, we can record when we manipulate the cell body. And now we're asking the question, what does the apical compartment do on the cell body? And so we can look at the effect of the disruption of the microtubules on the passive properties of the cell versus the influence of the apical dendrite. There we're seeing, unfortunately, we're seeing some effects at the let's say passive properties, intrinsic properties of the cell, but we do see effects of the coupling that are exceeding those effects. This is also a dose-response problem, and it's also a problem of how we're applying the drug, whether we're going intracellularly or extracellularly. It's also a problem of where we apply them in terms of puffing them, if we want to, at different regions of the dendrite, and timing effects, because some of these drugs take some time to manifest. So we bought ourselves a can of worms here, but nevertheless, it's really interesting. And I mean, in the final analysis, we're in the middle of this story, and I think it looks promising that we will end up saying that it has a disproportionate effect on the signaling of from the top to the bottom.
How would you explain the downshifting, the higher frequency going to low frequency from the top of the apical dendrite to the cell body, if it's not the microtubules? What in the membrane would do that?
When you say frequency, you mean back to what Earl was talking about in terms of oscillations? Or
aren't you saying that the frequency of oscillations of the apical dendrite slows down as it gets to the cell body?
So we haven't done, that's another experiment we haven't done, which would be really interesting. We haven't created oscillations and looked at their influence, which of course would also be, I think, affected by this. And of course there are resonant frequencies when you do that. So Dan Johnston, I think, has done a lot of what he calls chirp experiments, where you take every frequency and you give it to one part of the neuron. He normally did this to the cell body, and you find out what resonance the cell has, which frequency it likes the most. You can do the equivalent thing from the top of the neuron, give a chirp at the top and see what this does to the bottom, which we haven't done yet. That's just yet another experiment that would be on our agenda. Yeah. But I mean, in the end, I think we can get some way towards this. There's one other thing. I can see you want to embellish that question. Maybe.
Yeah, but I really appreciate that in a hardcore lab, physiology lab like yours, do this kind of dendrite and some microtubular experiment. And one of the things that I have been very curious during this conference is that Stuart's claim about megahertz, terahertz, gigahertz oscillation.
Yeah.
And I wonder whether you can actually patch clamp and then record this in a megahertz oscillation level of the voltage fluctuation under the very ideal kind of situation, and then interfere with the microtubule.
Yeah.
And if that just kill this megahertz, gigahertz, terahertz thing, doing that, then that's really like supports your idea, right? Probably. And yeah, probably you are the only one who can do this experiment in the world, probably.
Yeah, and to tell you the truth, we've just been doing what seemed to us to be the really basic experiment, and we haven't got to that. We're still, let's say, controlling the experiment. Eventually, that should be no problem, per se. So there is no problem for us to mess around at any frequency that our amplifiers can cope with. So there's a little bit of a problem in terms of the electrical properties at the tip of an electrode, but basically we can achieve this, I think. So yeah, that we should definitely look at this. Maybe the last thing I'd just say on this is that there's an interesting thing going on, because I think the OR hypothesis is is, I think, it's predicting something along the lines of dendritic integration theory in terms of, let's say, what comes out of the axon of the neuron. It's also making predictions that are kind of orthogonal, which are interactions, which actually Earl alluded to with ephaptic connections and so on, which are going between the dendrites. Yes.
That could be megahertz also.
Right, and these, that's where you might, so the interesting thing that I don't think, well, not quite sure how we would examine, are these, I want to say, orthogonal possibilities where things are going from one neuron to another without going through this loop of coming down the axon. So everything about dendritic integration theory is talking about the influence of the signaling from the top to the bottom of the neuron. Of course, it doesn't confirm or deny anything that would be going sideways, if you like, that not going through this pathway. And so I could imagine that in some larger sense, those sideways connections will be just yet another dimension of the causality of the system.
Like through the gap junction.
For instance, yes. It doesn't have to actually be a gap junction, because the electric fields are also going, if you like, from apical dendrite to apical dendrite. So, or gap junctions, yeah.
Okay, I have two announcements, but let's first give Matthew a big round of applause for... Thank you, that's great. So two quick announcements. Tonight, after the concurrence, after the posters, 10 o'clock in the restaurant/bar on the 13th floor, a round table with Nassim Harriman and Anirban Bandyapati, and it's a bar, I'll say no more. And then tomorrow night, the party will be on the third floor after the concurrence, and there'll be food, beer, and music, and from roughly 7 to 9, a poetry slam, and 9 o'clock onward music, and including dancing with our DJ Hide Saigusa, who's a great DJ. And if you're just in the poetry slam, please, we'll post a QRS or figure out some way in the morning, so you can think of the name of your poem, and everybody will probably, depending on how many we get, like 3 to 5 minutes each, and we'll do that from 7 to 9 and crown a champion by 9, and then dance. Thank you very much. See you at the concurrence.